Hey everyone, and welcome to this explainer. I am so thrilled to have you here today. Look, if you're a focused learner trying to really wrap your head around the mechanics of cellular life, you are in the exact right place. Today, we're diving deep into the fascinating world of epigenetics. We're going to focus specifically on that critical, super delicate balance between DNA and RNA methylation. We're going to unpack the science, look at the actual molecular mechanisms, and then explore the cutting-edge sequencing technologies that are making this kind of research possible.
So, think about this for a second. How exactly do cells control their genetic destiny without altering their underlying DNA code? I mean, your cells all have the exact same DNA, right? But a skin cell operates entirely differently from a neuron. How does the cell know what to do and when to do it? Well, the answer really lies with these invisible molecular regulators. They act like little switches and dials, constantly fine-tuning cellular functionality and developmental processes.
Okay, here's a quick look at our roadmap for today. We've got 1. The epigenetics of methylation, 2. DNA methylation, Genomic Archivist, 3. RNA methylation, dynamic regulator, 4. The epigenetic interplay, 5. Technologies unraveling the complexity, and finally, 6. Advanced sequencing by CD Genomics. Let's jump right in.
Starting off with Section 1, the epigenetics of methylation. We are looking beyond just the genetic code here. Let's define our terms. Epigenetics literally translates to above genetics. It refers to heritable changes in gene expression that happen completely independent of any alterations to the underlying DNA sequence itself. Think of your DNA as the hardware, right? Epigenetics is the software that tells that hardware exactly how to run. These chemical modifications, especially methylation, they fine-tune genomic stability and determine the cellular phenotype, all without changing a single genetic letter. It's brilliant. To really grasp how this works, we need to compare the two main systems conceptually. On the left side, we have DNA methylation. You can think of this as the stable archivist. It's basically the long-term reference library that keeps the genome organized and maintains strict cellular identity. On the right side, we have RNA methylation. Now this is the dynamic regulator. It manages the active, real-time cellular workspace constantly, and I mean constantly, adjusting to immediate biological needs. Moving on to Section 2, DNA methylation, our genomic archivist. Let's see how it maintains the master blueprint.
The crucial thing to understand here is the fundamental chemistry. DNA methylation predominantly targets what we call CpG dinucleotides. This is just where a cytosine nucleotide is directly followed by a guanine. A methyl group gets added right to the carbon 5 position of that cytosine, which creates 5-methylcytosine, or 5-MC for short. When this tiny methyl group is added to a gene's promoter region, it acts just like a tightly closed door. It compacts the chromatin and effectively silences that gene so it can't be expressed.
Now, the molecular machinery pulling this off is just absolutely fascinating. It's a precise two-step process. Step 1 is de novo methylation. So, during early development, enzymes known as DNMT3A and DNMT3B lay down brand new methylation instructions. Step 2 is maintenance. Whenever a cell divides, it absolutely has to pass its identity down to the new cell. So an enzyme called DNMT1, which is guided perfectly by a regulatory protein called UHRF1, comes in and meticulously copies these established marks right onto the new DNA strands. That ensures cellular identity is locked in and never lost. All right, let's shift gears to Section 3, RNA methylation, the dynamic regulator. We're looking at how the cell fine-tunes its workspace. If DNA is the long-term archive, RNA is the fast-paced factory floor. RNA methylation acts much more like a real-time switch. It dynamically regulates gene expression by basically deciding the fate of mRNA transcripts. It modulates how stable an RNA transcript is, essentially deciding, how long it gets to survive in the cytoplasm before being degraded. It also orchestrates how efficiently that RNA is translated into proteins. And while DNA primarily relies on 5-MC, RNA utilizes a highly abundant modification called M6A, alongside others like M5C and 2.0 methylation. To really bring the M6A enzymatic machinery to life, we need to look at its brilliant teamwork dynamic. This typically happens at a very specific sequence known as the DRAC motif. Here's how it goes. Step 1. A protein called METTL14 acts as a scaffold. It identifies the target and locks onto the RNA. Step 2, its partner METTL3 swoops in and acts as the catalyst, securely depositing that methyl mark directly onto the target adenosine. It is a highly coordinated biological tag team.
Which brings us to Section 4, the epigenetic interplay. Let's talk about this molecular conversation.
Here's the thing. These two systems, they don't just ignore each other. They're actually engaged in a constant molecular conversation. DNA and RNA methylation orchestrate functionality together. For instance, RNA methyltransferases, those enzymes we just talked about, they can actually alter the mRNA transcripts of DNA remodelers. And going the other way, DNA methylation can silence specific non-coding RNAs that would otherwise go out and regulate RNA methylation. It's this massive interconnected feedback loop that is completely critical to maintaining genomic stability.
When you actually compare these systems side by side on this table, you really see how brilliantly complementary they are. Look at the target residues. RNA heavily favors adenine for M6A, while DNA exclusively targets cytosine at those CPG sites. The core enzymes? Entirely distinct. You've got METTL proteins for RNA and DNMT proteins for DNA, and their primary roles perfectly reflect their nature. RNA handles that fast post-transcriptional regulation, while DNA is in charge of long-term transcriptional repression and chromatin structure.
Okay, Section 5. Technologies Unraveling the Complexity. High-resolution epigenomics.
So you can't exactly just look at a cell under a standard microscope and spot M6A or 5MC marks, right? The sheer complexity of epitranscriptomics, which is the study of RNA modifications, demands some seriously cutting-edge sequencing techniques. Identifying the exact locations and the downstream functions of these elusive marks is a huge challenge for modern molecular biologists. To get this right, you absolutely require a reliable, top-tier sequencing partner.
And that perfectly sets up our final section, Section 6, Advanced Sequencing by CD Genomics.
CD Genomics is basically the premier leader in RNA modification sequencing, and their solutions are perfectly tailored for this kind of comprehensive research. They offer this incredibly robust suite of services that directly tackle the complex biology we've just been unpacking. Need quantitative M6A mapping? They've got MeRIP-seq. Trying to find M5C with single base precision? They use specialized bisulfite treatment in their RNA BS-seq. They also offer RIP-seq to unravel RNA-protein interactions, 2O-methylated-seq for stability profiling, and even highly specialized sequencing for acetylation and pseudoridine. Literally, whatever epigenetic question you have, they have a specific sequencing tool for it.
Now, what I absolutely love about this next point is the accessibility. 5. That is the number of micrograms of total RNA required for their trace-level MERIP technology. Historically, doing this kind of research meant you needed massive sample inputs, but with a minimum requirement of just 5 micrograms, CD Genomics makes precise M6A analysis across mRNA, lncRNA, and circular RNA incredibly accessible, even if you're a researcher working with highly limited samples. It's a real game changer.
Plus, they offer state-of-the-art Nanopore RNA methylation sequencing. This service is amazing because it provides long-read, real-time sequencing of mRNA and non-coding RNA without needing bisulfite treatment or even reverse transcription. By skipping those steps, it allows researchers to capture these incredibly subtle nuances in RNA modifications with heightened accuracy and true single nucleotide resolution.
Which brings us to the end of our explainer, and I want to leave you with one final exciting thought. What hidden epigenetic mechanisms will your next research project reveal? We've just seen the intricate dance between DNA and RNA methylation and how they jointly govern the molecular workspace. With the advanced tools and services provided by CD Genomics, your next major breakthrough in understanding cellular functionality is entirely within your grasp. Thank you so much for joining me for this explainer. Keep learning, and I'll see you next time.